And thank you to everyone that is listening to us today. It's a huge pleasure to be part of the Basics of EEG Technology lecture. So this will be, like Christoph said, something that we will approach the basics of EEG. So it will be a lecture more related to people who are not so experienced, but also to the ones that are experienced. It's always a good reminder of all the best practices so that you're able to acquire EEG at the best quality. My name is Francisco. I'm working for g.tec since 11 years. And I'm an international sales agent. Yeah, so today we will talk a bit about this particular topic. Uh, in the program today, we will approach a lot of things, and we will be able at the end of this 1 hour to answer all these questions. First, what is an EEG and what it is for? Second, how did the EEG devices develop across time? So we will have a look at how g.tec was also part of this EEG device history and how important EEG devices can be for your EEG acquisition. Which are the requirements to acquire the best quality of EEG? So going a bit from what is the best setup to how to acquire with the best quality of signal. What is an electrode montage and why are they important to your setup? And also talk a bit about what kind of electrodes exist and what are they best for? Which purposes do you use this particular electrodes and for what purpose do you use the next particular electrode? Then we will do a practical session where we will have a couple of colleagues showing how to set up and compare between the usage of passive and active electrodes. We will also have a comparison between wet and dry electrodes, because I've seen that in the chat there was a lot of questions about it, so I hope that I'll be able to answer them all. And then also a setup in comparison between fixed and wireless devices. So we use fixed devices, wireless devices, active electrodes, passive electrodes, wet and dry. So we will also have, if we have time, hopefully, we will also approach one of the biggest and most important things that everyone should do, which is the cleanup and the maintenance of the devices. So just to start, what is actually EEG? So here what you can see in the screen is the first human EEG traces recorded by Hans Berger 100 years ago. As you can see, he had a very big devices with 1 or 2 electrodes, which were able to first detect the first EEG recordings. So EEGs are actually electrical potentials that we can see, namely for EEG on our brains. Typically, you will put EEG electrodes on the surface, so they are surface electrodes, they're not invasive. You will put them into the head and you will try to see what kind of electrical changes are occurring across time. There are different imaging techniques. We can see here on the top a very well descriptive image of what or where EEG is placed. EEG has a very big temporal resolution. This is the highest important thing. So you're talking about differences of brain activity detection on milliseconds, which is a very big thing compared to, for example, fMRI, where you're talking between seconds to minutes, or PET minutes to hours. So EEG can detect very quick changes of brain activity. And this is something that we will see here today and that we will see also in the different lectures across these 10 days of spring school. One other thing that is important to know is that the EEG has a special resolution, which is, for example, not as great as the fMRI. So we have an electrode which sits on the top of the head. You have a lot of different layers between your electrode and brain. So what you will be recording is a couple of centimeters around the area where the EEG electrode is placed. This will be, for example, much better if you use ECOG. So if you implant grids inside, so you get rid of the hair, you get rid of the scalp, you get rid of the bones, and you have the electrodes exactly on the top. And obviously then the spatial resolution will be more for better for ECOG compared to EEG. But what is the best part of EEG is that they are not invasive. It's easy to use. You can use it everywhere that you want. So it's a very versatile kind of imaging. And this is the biggest reason why it's so widely used for neuroscience and widely used for neurology research. What kind of things we are looking when we are recording EEGs? So you have seen also in the previous lecture, Martin talking about the frequencies, how we're going to see different frequencies on the EEG. You have seen how by closing his eyes, Sebastian was able to induce some alpha activity on the screen. So you see this higher amplitude EEG traces. So this is something that many of the studies, for example, long cognitive studies where you are putting subjects through paradigms where it requires them to relax, then maybe do some cognitive task, move different parts of the body. So you'll be able to discern between different kinds of frequencies which are happening in a short period of time. Another very important thing that you can reach with the EEGs are obviously the evoked potentials. And here we're talking already about the temporal resolution where EEG is superior to most of the other images. You can detect very tiny changes on our cortex just by having an evoked potential or having an event marker and measuring how much time after this event marker, can be a stimulation, can be a task, how much time our brain takes to react. And these traces are usually used, for example, in BCIs, where we've seen the unicorn being able to discern, for example, faces versus other images, or selecting items for video games. So you can use these kinds of paradigms to be able to discern different kinds of brain activity and what kind of activities the person is using while wearing an EEG cap. So how did the devices develop? So you've seen this image on the left is showing one of the first EEG devices. So they occupied a large room, so you needed a very big room to have an EEG devices. And you have seen today the introduction of the BCI core, which is a device of this size. How did this happen and how long did it take? So as I said in the beginning, the devices were very big. 100 years ago, we started recording the EEG, and g.tec is also part of this history of devices and development of hardware evolution. So you can see here a picture where Christoph and Günther are very young, showing one of their first devices. Also here, Christoph with very long hair. Also lack of gray hair on both of them. So you can see how big the devices were in the past and how we were able to reduce its size to the bare minimum. So this is the first device that g.tec presented in the conference. Christoph already talked about it. You needed the floppy disk. You were connecting to a DAQ board on a very big CPU. This is the preliminary hardware development of g.tec, which after some years, we developed the BSAMP, which is a pure passive electrode. You also need a DAQ board inside the computer so that you can connect it directly to it. You record 16 channels max, passive electrodes, and that's very simple. There's still people using today this amplifier, so it's like a war machine. It doesn't get destructed. And many people really like passive electrodes and passive EEG recordings because of how low the impedance, how low you need the impedance to be so that the EEG can be actually visualized, and that obviously also stabilizes the EEG across the whole measurement. After that, we developed the G-USB amp. Very similarly to the BSAMP, it also had 16 electrodes, but the biggest difference are that not only you don't need a EQ box anymore, you can just connect it via USB, therefore the name USB amp. You also can start recording with active electrodes. So electrodes that typically have a preamplifier close to the source, making it possible that you record the EEG without needing to have such a low impedance as for passive electrodes. And after some time, we saw the need to develop the G-high amp, which we will be using today. G-high amp, very similarly to a USB amp, you connect it via USB to the computer. It has a high sampling rate of 38 kilohertz. You can record active and passive electrodes at the same time, and the biggest difference is the number of channels. So you can go up to 256 channels on the G-high amp, and for the USB amp, as I said, 16 channels. What happens also in the other sector, in the wireless sector? So we were the first company to have a commercial wireless EEG device, which was called the Mobilab. So it was very, very tiny, small device, which you can put it onto the pocket. You had 4 AA batteries, which are powering it. You could record up to 8 channels. You had the possibility to record passive and also active, so using the same technique as the USB amp. And you had the connection via Bluetooth to the PC. So the distance between the user wearing the device and the EEG cap could be way apart without having the need to have cables in between the two the user and the PC. Then after that, we saw the need to have an even further device that could even enhance the signal quality and the number of channels. So we developed the g.Nautilus. You can see here one of the g.Nautilus research. They can go up to 64 channels. The sampling rate can now go up to 500 channels, up to 32 channels, 500 hertz. You can record active, you can record the dry and wet electrodes with it. And the biggest change is that you have the connection via Wi-Fi. So we stopped the Bluetooth for the research devices, and we have a Wi-Fi protocol that doesn't allow you to have interference from other devices, as only the receiver can read this amplifier, which is sitting on the back of the head. Then, because people are not using only EEG for recording, right, in their labs, so they have the need to add sensors, to add EMG, to add ECG. We developed g.Nautilus research multi-purpose. So the only big difference is that you have connectors, so you can disconnect one EEG electrode, and with a 1.5 touch-proof connector, you can just input sensors and other kinds of electrodes for EMG and ECG. So the user can be far away, up to 10 meters indoors from the PC. There will be obviously cables because you're adding sensors, but the sensors will be connected to the amplifier sitting on the back. And you will be able to have a completely seamless wireless device for not only EEG, but other kind of sensors. And then the last development was the g.Nautilus ProFlex. g.Nautilus ProFlex is very neat. You have a very small amplifier, which adds a connector on the top, and then you can have different caps. So this amplifier goes up to 32 channels, and then you can have different caps with different sizes, different electrodes, different kinds of electrodes. The opportunities are endless. And they allow you to, for example, if you are planning to record a lot of subjects across the day, you don't need to have an amplifier connected to a cap and wash it and then let it dry. You can just measure the person, then remove the cap, remove the amplifier from the cap, connect it to the next cap, and record the second person. So it's very, very it's widely used for users that want to measure a lot of people in the same day, but also users that want to try different montages, try different kinds of electrodes, etc., etc. And then we entered back in 2019, we entered the consumer-based device sector with the Unicorn Hybrid Black, which you have seen on the lecture before. The Hybrid Black has 8 hybrid channels. And now we developed also a second version of the Unicorn, which is the BCI Core, which not only allows you to have a much more flexible changing of caps comparable to the Unicorn Hybrid Black. It also allows you, for example, to record animals, to have a very good research package with Python APIs and gpipe. You'll be able to hear about these more in detail in more dedicated lectures in the next days. So most importantly, what do you need for the perfect EEG recording setup? As you can see here, this is one of the many opportunities that you have for a complete lab. Not only you will need a EEG device with a cap, but you also may need other kinds of systems, other kinds of amplifiers, other kinds of sensors, other kinds of stimulators. So you can see here a very complex setup where you have a subject typically sitting on an armchair, an amplifier close to the subject if it is a wired device or a tabletop device, like I like it to call it, in front of a screen, which is showing some paradigms, with a recording PC, with data storage, etc. So it really will depend on what kind of applications you're doing. For example, do you want to do stimulation of the user so that you can detect different changes and modulations on the EEG? Then you may need, for example, a stimulation device like the G.tec box, where you can apply auditory stimulation, tactile stimulation, visual stimulation, etc. Do you want to have the subject to have, for example, an interface with a fast stimulator, which will actually perform the movement of the hands? Christoph talked in the first lecture, so you will need a eStim fast. Do you want to do VR applications where instead of having a screen, you have VR glasses? Everything is possible. So it will really depend on what kind of applications and your main goals are. But what is most important is actually selecting the best way to record EEG. EEG devices are very neat, but they can be very, very sensitive to the ambient. So having the correct environment, for example, having good grounding, not only on the building, but on the subject. Sometimes the person is using different kinds of clothes, which will induce static charges. This will be very important on the dry electrodes. So selecting the correct environment, so good grounding, isolation from interference from other electrical devices that are communicating in the same room is essential. The correct setup is also very important. So do you want to record a highly extensive paradigm where requires the subject to be highly concentrated? Then a quiet place will be essential. You will have to have a lot of things to take care of. For example, one of the biggest problems that I have, for example, when I go to a conference are carpets. Hotels like to have carpets, but carpets are not really friendly to dry electrode setups, because they can create static charges and its usage can induce some artifacts on the dry electrode system. So having the correct setup can be very, very, very important for having a very high quality EEG recording. Then you also need to have a well-maintained device. So you need to have electrodes that are thoroughly clean. So you typically, when you use a gel electrode, you will want to have at least at the end of the day to clean it thoroughly so that there is no possibility that the gel that was applied to dry and create a dry layer on the metal parts, which will then complicate the EEG recording. You will also have to check if the device doesn't have any current leakage or damage, so that you have a very secure and well-maintained device, so that your recordings are on spot. You also will have to have the appropriate tools for the different types of electrodes. So for example, for passive electrodes, you will leave abrasive gel. For active electrodes, you will need electrode gel. For example, if you use some setup with some sponges, you will have saline solution that you can use with that particular electrode. So each different type of electrode will require different tools, and you have to take care that they are actually as good as possible and as tested as possible. Always check with your manufacturer to see if the abrasive gel or electrogel or the saline solution you use is appropriate for this type of electrodes. Another thing that you will want to take care is the correct size of the cap. So different people have different head sizes, and it can be that someone is using a very tight or a very loose cap, and you will have electrodes floating around and not really touching the scalp. This will complicate not only the setup, but also the way that you record EEG. And obviously, because of the cap and because of where you're putting the cap, you will need to know if the electrodes are placed on the correct spot, especially in particular montages where this is highly important. So a good placement of electrodes is also essential. So you have to verify if the positions are well placed on the scalp. And there are several techniques, and we will show you today a couple of them. Exactly. Then you also need to have appropriate appliance of tools. So you have to make sure that you have enough gel to reach the scalp, that you use syringe to push away the hair so that you can really reach the skin and then start applying the gel. You have to verify the signal quality. You can check, for example, the impedance values for the wet electrodes, check for artifacts. You saw today Martin telling Sebastian to clench the teeth or blink the eyes, closing the eyes and relaxing to see the alpha activity. These are some of the verification techniques that you have for the signal quality. You have to ensure that there is a good connectivity between the amplifier and the computer, obviously, not only for the wireless, but also for the wired, so that everything is fine. So if you have difficulties in connection with the wireless device, you will have flat lines, you will lose a lot of samples, and this is less than ideal for your recordings. And you also have to ensure that the signal quality maintains across the whole time that you're recording, especially if you're doing, for example, overnight recording of sleep studies or 24 hours EEG. Not only that, but also in terms of measurements of 1 to 2 hours, where you need to know that the quality of the signal at the end is as good as in the beginning. So the dropping of quality of the EEG can happen because you put too little gel. The gel is not appropriate for this length of recordings. You have, for example, problems on the connectivity of the dry electrodes onto the skin, etc., etc. So you need to make sure that the signal is as good from the end as in the beginning. And usually the best requirement for the EEG recordings is actually to have the possibility of visualization of the EEG signal in real time. With real-time EEG visualization, you will make sure that what you're recording is not pure noise, pure garbage. This is highly important because you can imagine you start recording, the signal is good, you don't have visualization in real time, or you're just checking raw signal. Then at the end, you stop the recording after 1 hour and a half. Then into the next day, you go to your offline analysis tool, and then you realize there is no EEG there. There's just noise. The signal is completely bad. So having a real-time visualization of the EEG can be highly important for that, so that these errors do not happen. So what is an EEG montage? EEG montage is the positioning of the EEG electrodes on the head. Uh, what all of you probably already know is the International 1020 system. There are other systems as well, which measures the distance between the inion, which is this bony structure on the back. I don't have hair, so you can see much better, to the nasion, this bone between the eyebrows. These are the first measuring points vertically, and then you also have the left and the right preauricular points. And on the top of them, on the intersection of both of them, is the position of CZ. And from that point, you will measure all the other positions as well. We'll be able to see how to measure it today. And we will also have the opportunity to use the caps of g.tec, which already have this pre-positioned electrode recorded or printed on the cap. So why are EEG montages important? For example, you want to do a measurement for evoked potentials, visual evoked potentials. Which montage will you use, and where will the most of the electrodes will be placed? Obviously, if you're doing a visual evoked potential, then you would want to have all the electrodes, or most of the electrodes, the most important electrodes, on the visual cortex, because that's where most of the changes of the activity will occur. For example, you want to do a motor imagery, like we mentioned in the past, then probably the visual cortex is also important, but not too important for this kind of measurement. So you can have the electrode montage placed on the top, on the motor cortex and the somatosensoric cortex, so that you have a EEG recording on the exact spot that you're trying to measure. So having the correct montage, the correct number of electrodes, and the correct position of the electrodes can be very, very, very important. So let's talk a bit more about the electrodes themselves, right? So as you've seen in the beginning, we were using passive electrodes. We have developed in the past many different kinds of passive electrodes. Here on the top, this is something that is widely used still today, is the golden cup electrode. So you have a golden cup. You will put the cup into a gel, and then you place it into the head. You have to measure, then you put a tape on, and you measure the EEG. So this is passive electrodes. You can still use it today, and there are many applications which you will see, for example, for EEG for horses, where this totally makes sense. Then we had some screwable passive electrodes, which were the first caps of EEG that we had. We had the G butterfly, which was already an active electrode, flat one, which you could put on already on the cap. You don't need to have any kind of tape to record it. And these are the currently commercially available electrodes. You have the G Ladybird active. So you have around 1 centimeter of measurement of the electrodes. They're a bit flat, which allows you to do a lot of different applications. Then we just released last year the G Ladybird passive for TMS. So these are the flattest EEG electrodes in the market. These are typically used for TMS recordings, for also passive and active. So you will be able to see these recordings on the lecture in the next days. Then, because people wanted to record more and more channels on the scalp, we developed the G Scarabeo electrodes, which are a bit tinier than the G Ladybird, and allows you to record more channels on the subjects. Then we also have the G pangolin electrodes, which we released in 2020. So these are high density electrodes. For it, you will need a head like mine, so freshly shaved. And these electrodes have around 2 to 3 centimeters, and they are able to record 16 channels. So the resolution is very, very big, especially compared to the other EEG electrodes. And there are a lot of applications and a lot of very, very interesting projects which have been used. G pangolin, which could find things that typical EEG electrodes could not find. For example, the difference of the movements of the fingers, which the EEG electrodes have difficulty in capturing. And then we also developed the first dry electrodes, the G Sahara, which are these ones on the left side. They are silver chloride, golden plated. They, instead of applying gel, they have pins which go through the hair and then really reach the scalp. And in that way, you can have dry recordings without the need of applying gel. And in the past years, we also developed an update of the G Sahara, a G Sahara hybrid. So they are made with a polymer compound. They are hybrid. This meaning that you can either do dry recordings, or you can also apply a bit of gel by having inserted a bit of at the tip of the syringe from the top, and then applying a bit of gel. These are widely used, for example, on the unicorn, but also for research. And what is actually the difference between the active and the passive electrode? So you can see a bit here what kind of signal you will be able to see. So the biggest difference is that you have a preamplifier, which is activating the electrodes and allowing you to have a higher quality of signal without needing to have such low impedance. So for example, active electrodes require no preparation or abrasion of the skin compared to passive electrodes. They also have less 50 to 60 hertz noise caused with the high impedance. And they have reduced artifacts from electrode and cable movements because the preamplification is typically occurring on the electrode itself and not just at the amplifier side. What is the difference between gel and dry? So obviously one has gel, one does not have gel. But what is actually the drop off of this? So for dry electrodes, because you are not having any kind of gel, the impedance is even higher. You will have to increase the sensitivity of the amplifier, meaning that you will get a very good EEG, but you're also able to get other kind of artifacts, namely, like I mentioned, from static charges. Still, this is a very widely used electrode setup because not in all the studies you're able to actually have a lot of people being wanting to measure with gel electrode because they don't like to put gel on the head, or because you have so many people in one day that preparing so many people will take a lot of time. What we did with the dry electrodes is actually trying to test it between the gel and the dry electrodes, and we found out in the views already more than 15 years ago that we actually have pretty good signals. So applications like evoke potentials, frequency band, you can actually detect very good with the dry electrodes, which allows you to use these kinds of electrodes on your setup. So we can see here, for example, how the accuracy changes, but you can see that the dry electrode, even though it's not as good as the gel, is highly significant. So we'll have a lot of people who will be able to use dry electrode setups on their experiments. So let's go to the practical session. What will we use today here? So we will do recording of passive versus active electrodes. We'll be using a G.high amp, which is the high amount of channels device that we have here. We have two G.high amps, both with 80 channels. We have one person who will have a cap with 4 Ladybird passive electrode channels. We will have another person with 16 G Scarabeo active EEG electrode channels. We will try to see how different, how they score in the speed of preparation and how the signal will look like. And also we will have a look at the values of the impedance. We will use the same software, the G Recorder Pro. The difference of the accessories needed are then on the passive, you will need to have preparation of the skin, abrasive gels, cotton buds, conductive gel, and syringe. On the active, you will just need a syringe with gel, and that's it. For this sample, for this demonstration, I am using something that on the last year people complained a bit because what if I'm super familiar with passive recordings? I'm sure that I can do as fast as active. So I picked 2 colleagues. One of my colleagues is doing passive measurements almost daily, and he will be using the passive electrode setup. And I have another colleague of mine who is actually the main person responsible for G Recorder, who is not very familiar with applying gel into the electrodes. So let's take him as an example of an unfamiliar user, and we will be comparing the speed of both of them. So let's make a small competition. This will be the first demo. Then we will show you the active electrodes with wireless setup and the passive electrodes with the setup. For this one, I wanted to show you a bit of how easy it is to change the amplifiers with the G.Nautilus Pro. So we'll have someone having a G.Nautilus Pro with passive electrodes, 16 G-Sahara hybrid, and another person with the same person will be switching then to active wet electrodes, so 16 G Scarabeo electrodes, and we will be able to see that without changing anything on the computer setup, we'll be able to record and switch caps pretty easy. That's it. I will now change to the other webcam to see the setup that we currently have. And yeah, so let's go. I will also show you now. Let me close the presentation, also show you the setup that we are having here. And I will connect now the amplifier just to show you how easy the G recorder is. Let me remove one of the, okay. So I hope you can see my screen. I'm trying to see where my mouse pointer is. Ah, there we go. So let's connect on G recorder. We select the hardware. The G recorder is trying to find the amplifier. I need to turn on the amplifier before. So I turned on the amplifier. I heard the Windows noise that the device is connected, and he will be able to find the connected device. So here we have, you can see the serial number here, and you select the hardware and press OK. So everything should be set up. We can go to the G high amp. We are recording just 16 channels, so we can deselect all the channels and then go from 1 to 16 and select it here, where the channel 16 is actually the reference. So let's select it here. In this setup, we are using a typical setup for TMS, where the reference is not the earlobe, but in the front, just so that you know how the setup looks like. So we will not be applying any kind of filters. And I will load the electrode montage, which should be the same as the Motor Cortex MI. And then you can see now on the channels, not only the number of the channels, but also the positions of the channels. This will be very important because we want to have an overview of the impedance check also in 2D. So I will open the impedance check. Impedance check is on the other screen, so I'll just drag it here. And then you can see here the impedance values of all of them. So you can have in 2D, in 3D topographic, or in 2D flat. And you can select the type of electrodes, active versus passive here. And yeah, that's basically it. I will shift and stop sharing the screen so that you can see just from the images the difference of the let me just stop sharing so that you can see the camera. I will actually put the camera on the other side. So there we go, everyone is here. Please stand up. I will just maybe put the camera on the back so that we can see. On the second PC, can we do the same already? To put the G recorder, I'll just drag the PC here. Let me see the camera that we can see. Oh, yeah, it's okay. It's okay. It's just, can you open the, so we can see on the other screen, it will be hard to see, but Slobodan is going to put the 4 electrodes, so 3 electrodes, so ground, ref, and 3 electrodes, and everything is already selected. So now let's open the impedance check. So same way that I did is closing the configuration and now going to the tools and impedance check. So we will have the impedance check of the 3 values. And we will have, thank you, Christoph, for closing the shades, because in the morning it was not so bright as now. And yeah, so I will ask both of them to start applying the gel. So we have Matthias here with the active electrode system, Slobodan with the passive electrode system, Alex being the subject of the active system, and Clemens being the subject of the passive system. So I'll put the camera here. Let me just look at so that I can also see the camera. So let's go. 1, 2, 3, let's start. So Matthias starts by putting the cap into his head. And Slobodan is preparing first the skin. So you can see he's very, very efficient on it already. So Clemens has a bit of hair, so he needs to clean all the scalp. So he's removing first with alcohol so that you can remove a bit of the fat, natural fat of the hair, dead cells. In the meantime, Matthias is already measuring the head. So the cap is already on the head. He's measuring the distance between the anion and the nasion, checking if everything is fine. All good. And is now starting to apply the gel. In the meantime, Slobodan firstly puts the cap into Clement's head. And we can already see Matthias putting the first drops of gel into the head of Alex. You can see how inexperienced Matthias is. And you can see how experienced Slobodan is. I think Clemens cannot see much because his cap is almost covering his eyes. But this is already on channel number 4. We can start the, let me just start the impedance. So we can already see results there. Some of the channels are already looking good. So then can you start impedance so that we can see? Yes, but we want to see the speed. So Matthias is just putting a bit of gel. This twirling motion is important to really reach the scalp. Slobodan is still searching for the movement. So he's using now abrasive gel. So he cleaned the head from any potential grease. And now he's applying very strongly abrasive gel. You can see already most of the electrodes in green on the active electrode setup side. Montage is really nice because if you're completely lost, 16 channels is easy, right? But if you have 64 channels or something like that, and you're completely lost which of the electrodes is bad, then having a montage on the impedance check is highly important. On the passive electrodes, obviously we are using just 3 electrodes, so it's not highly important, but you can still put the montage if you want. You have to do it like I did it on the past. So Matthias is almost complete. Maybe we can improve a bit some of the channels. So you can see most of them are already green. On this side, not much changed. It's fine there. Yeah, just put it there. Very, very cool. So all good. We can see already that someone using the active electrode is having much more success compared to someone using the passive. So finally, it reached the time where Slobodan is putting conductive gel, after putting the abrasive gel, after preparing the skin, after irritating the skin in order to have a lower impedance. And we'll be looking at how they measure. So you can see all green on this side, all green on that side, but the difference is abyssal in terms of preparation. Never forgetting that we prepared here 16 electrodes and there just 3. But it's slow, so Slobodan, you can, this is proof that Slobodan is very good at passive electrodes. I can see now low. and also low for all the electrodes of the active electrode. We can, if I may, pass here to, no, this is good. You did a good job, thank you, Matthias. How many times did you measure before? How many people in your 40 years of life? 10 in 40 years seems little. So we can see a lot of, let's apply the 0.5 to 30 Hertz and not filter. Let's go to the EEG channels. And we can have already here vision. Alex, I'm moving a lot. Can you blink? You can see the blinks there. Can you close your eyes and relax? I can see good alpha activity there and clench the teeth. Yeah, works good. Let's go to this one. I'm moving a lot, so our floor is a bit bumpy. Clemens, can you blink? Yeah, you can see the blinks there. Can you clench the teeth? Yeah. And finally, can you close your eyes and relax? Oh, half activity, very nice. You need a coffee, Clemens. So you can see here the difference of setting up between the two. 4 channels, you needed almost as much as 16, but you needed to do a lot of steps in between. For active electrodes, even someone who is not highly experienced can just follow our setup of measuring the impedance and looking at the values of the colors and be able to have very good recordings of EEG. Okay, I would like to thank you, our guests. I will stay with Matthias because we will go now to the recording of the wireless device. So I will just place the camera here and maybe we can sit there, Matthias. Sit where I was sitting. Yeah, thank you so much. Thank you, Alex. Okay, so there we go. So let's measure. I will show you a bit of how to set up the wireless system. So we've seen that with the wired device, you will need a lot of different kinds of cables, and you saw how the tables looked like. This is all you need for the wireless electrode. This is the amplifier. This is the G.Nautilus Pro, which you can just press the button for 3 seconds until the light drops, and then it should start blinking fast, searching for the connector, for the receiver. The receiver is here. So you can see the receiver here. You have a port which you can send digital inputs directly into the amplifier, which will be recorded on G-Recorder as event marker and MATLAB as an extra channel. And then you have the connection via USB, which I am going to do. I just pass in front of the camera, which is not ideal, but there we have. So between Matthias here and the computer, there is no cables at all. And we have here today 2 different caps. We will start first with the cap of the hybrid electrodes. So you can see G-Sahara hybrid, which is something that you also saw on the Unicorn. And in this version, like I mentioned, is the multi-purpose version, which you have a connector, which you could connect, for example, different kinds of applications like sensors, EMG, ECG, whatever. And then we have the other cap, which is the same that we use for Alex. So it's a G-Scarabeo cap. They have the connectors here, which we will connect to the amplifier. And now we will jump to the preparation of the dry electrodes pretty quick because time is running. So you saw the difference of the preparation of the other one. So we will have a cap connected to the amplifier. So I'm measuring, like I said, for the montage, the difference between the anion and the nosion. And the CZ should be then a bit more to the front. Sorry, Matthias. It's good enough. So we should be the CZ here is fine. Then I will just roll around because Matthias has a bit more hair than me so far. And we will use 2 stickers, which are here. Okay, thank you. 2 stickers for reference and ground. So a bit something like the unicorn. Behind is here. And then just close the cap and start measuring. So I'll also use the G recorder so that you can see the same setup. There we go. Let me connect to the G recorder. Okay, let me go back to, yeah, thank you. Yeah, we turned off. I can start sharing the screen as well. Let me see, share the screen. Good. We will do the same thing. We will just press the setup, settings, select hardware. The device is blinking, so it should be able to connect. We can see already the G high amp disappeared. We add the G Nautilus, we press OK. We will then go to the settings because we are having a passive electrode here. We will change the input range to a higher setup. Whoops. We will apply, and then we can also already apply because we don't have too much time. 01 to 30 Hertz and notch filter 50 Hertz. And that should be good enough. I will maybe increase the channel view to 70 so that we can see good the artifacts instead of 50. And then we press just start. And we should be able to see, let me increase the number of channels. And good. We have here the device connected. And we have the connector here. And we can connect the device, put it into Matthias cap. And that's basically it. Now we wait a bit to see the EEG signal there. Can already see something. Very good. Matthias, can you blink? Can you bite? I can see the bites. And let me just, there is just one electrode, number 11. So to have a very good connection to the skin, you will need to twirl a bit the electrodes so that they really reach the scalp, right? So that the hair just goes through the pins and then really reaches the skin. So it's a bit better now. Just this channel here. And let's see if the letters on the back, they're also good. Just this 11, which probably is one of the connectors which you have to put better. Yeah, so you can see very easy to set up. So you don't need to put much effort into it, and the signal is already applied and recorded without needing of any extra appliance of gel or anything. Quickly, 4 minutes, just let's switch the cap, but not the amplifier. So the amplifier remains the same. It's this one. We will take the cap, the passive, the dry cap from Matthias, and we will now start to make him wet. So I will put, you can see on the screen, the same setup, same everything. Same user in this case, you could have another user. probably is better because then you're quicker into setting it up. I was a bit aggressive because I like being aggressive with Matthias. And we have, in this case, the gel. In this case, we have the syringe. I'll apply a bit of gel into the earlobe reference, applying a bit of gel in the ground, and then one by one, a bit of twirling around so that the tip can reach the hair. Let me see. Obviously, like usual, you need to connect the amplifier to the head. In this cap, sorry, but yes, this is not good. Now it's more comfortable. We have a magnet holder, and then we can see already the signals getting better. Whoa. I need a signals getting better one by one. You can see you can, with the same setup, same amplifier, same software, you can change from one dry electrode to an active electrode without any issues at all. With this one, you can also check the impedance like we did, but most importantly, what I wanted to show is literally how easy it is to set up and how easy it is to have all the electrodes being placed together. You can see already them coming back. This is channel number 10. They are number-rated here, so we can apply a bit more gel if we find it necessary. So you can see already 10 is good. Let's see if he comes back. The filters are applying, so it takes a bit of time. Now it's good. Can you blink with your eyes, Matthias? Good. Can you bite your teeth? Good. Can you close your eyes and relax? Oh yeah, we can see some alpha activity. And you could see it's pretty easy to set up as well. There is no cable, so Matthias, maybe you can walk a bit. I will always, so you can see he's moving. There is no lies. There is no cables here between him and and yeah, so you can see some of the artifacts because he's moving, he's doing some martial art. So very, very good. And basically that's all I wanted to show you with our systems. And I'm open to any questions. I will change the camera to a more appropriate angle of mine.
Frankie, thank you very much. I will start with a very difficult question right away. What's the difference between mu and alpha waves? Let's see if you know it.
Yes. So alpha waves are lower frequency waves, which you will find especially on relaxation mode, closing the eyes and trying to relax. Mu activity is something that we typically find, for example, when you're trying to do performance of movement of the limbs, is an activity between alpha and beta, which allows you to detect, for example, correct CSPs, meaning that you are able to detect if someone is trying to actively imagine or perform the movement of one of the limbs or the other ones, if you are particularly focusing on electrodes on the motor cortex.
Very well. So mu is sensory motor cortex activity, sensory or motor. And David wants to know, are the dry electrodes uncomfortable after a longer time?
It will really depend. So depends also on how much hair you have. So for me, it's because I don't have any hair that is damping the electrodes. After many hours can be uncomfortable. But you can see the new kind of electrodes. They are very flexible, so they fit very good in the hair and the head, and they don't provoke a lot of uncomfortableness. So after changing to these new electrodes, I have stopped feeling all the pain on the golden electrodes. And I truly recommend it because they are very neat and they are easy to use.
Daria wants to know, can you put on the electrode cap with the gel onto yourself?
Into myself, yes. You can typically put it into your own head, and you can use a mirror, or you can use, if you're experienced, just go with the hand and then one by one. If you have, for example, like I showed you, the impedance check with a montage already in 3D or in 2D, you can easily check which of the electrodes are used. Obviously, putting it into another person is much easier because you have an overview, but it's also possible to put it on yourself. I have done it in the past and worked pretty good.
So we reached exactly 11 o'clock. Thank you very much for the nice competition. People liked it. Very well done, as usual. Thank you very much. And